All right. Hi guys. We're gonna get started with topic four. Topic four is what's it called? The like transcription factors, trip operon, and then at the end of topic four, I think it's like non-coding RNA. So that's like mRNA, sRNA, and stuff. We're not gonna get into that part. That'll probably be covered more in Thursday section, just because I feel like beginning half of the week, not a lot of people get to all of it. If you guys do end up having questions on it, obviously we can answer it. We're not going to say no to helping you guys understand it, but let us know. All right. Oh, I'm Tina, by the way.
Oh, and I'm George, right there.
So we're going to start off with transcription factors and why we have them. So transcription factor mediated transcription regulation. I think last week we talked a lot about like different types of regulation, but this week we're going more specifics into like operons and transcription factors and how they play a role in operons. And so transcription is from DNA to RNA, but transcription factors, they control RNA polymerase binding to the promoters in genes. So if you didn't know, we're going to go into this as well, but promoter is where the binding starts, where the polymerase binds to on the gene, and then the transcription can actually happen. So RNA polymerase would bind to the promoter and then actually create the RNA from the DNA strand. But the transcription factors are what controls it. So you can either have general transcription factors or regulatory transcription factors. The difference between general and regulatory is that typically general transcription factors are not sequence specific. So that means like they can bind to a lot of different places or a lot of different DNA strands, whereas regulatory transcription factors tend to be more gene specific. If they bind to like this part of the gene, you're not going to see it at like another part of a gene just because that's they won't be able to bind to it. There are like some like caveats as usual in biology, and it's never like so straightforward. For example, sigma factor, which is in prokaryotes, technically they are transcription factors, but they are also sequence specific. And then that's going to be on the next slide as well. But for regulatory transcription factors, you can have positive or negative regulators. So if you have a positive regulator, you enhance transcription. So that means it's going to try to help RNA polymerase bind more often, and then you'll have more transcription. Or you can suppress transcription by being a negative regulatory factor, and then that means that it might like prevent RNA polymerase from binding, or it might block RNA polymerase from doing what it has to do. My laptop's a little slow, sorry. It's gonna take a second.
Wow.
I know. Yeah. There you go. Okay, so like I was saying, sigma factors are in prokaryotes. They're also in eukaryotes. Before this class, we tend to talk about prokaryotes anyways. So you don't need to worry too much about how they work specifically in a eukaryote, but they do exist if you want to know. Specifically, we focus on sigma 70, and it's a protein that helps RNA polymerase bind to the promoter because it is a transcription factor. It's a general transcription factor, like I said a second ago, but it is consensus sequence specific. So it binds at the -35 box, which is here, and then the -10 box over here. And then when it binds first at the -10 box, that's where the unwinding starts, because where the most A's and T's are. If you know a little bit more about like the chemistry, A's and T's only have 2 bonds, whereas G's and C's have 3 bonds. So 2 bonds are easier to break than 3 bonds. That's why it typically starts from here to start breaking apart. To explain a little bit more about the whole like sequence specific, consensus sequence, all that, because it gets a little bit confusing for people or for students. A consensus sequence just means like this is the best match possible. So sigma 70 can bind to a lot of different places, even though it's like -35 and -10. It is still general transcription, so it can bind to a lot of different places. However, there is like a consensus sequence where it binds the best. So like at the last bullet there, while it is sequence specific, it regulates a lot of different genes, so it can bind to various sequences, but the affinity can change. So if it happens to go somewhere where it is the consensus sequence and that's the best sequence that it can bind to, it's going to have a strong affinity. However, if it goes somewhere where it can also bind to, but it's not the consensus or close to the consensus, it might still bind to it, but it's just not going to have as strong of an affinity. Does that make sense? Okay, cool. Yeah, so then sigma binds to the DNA, and then sigma binds to the gene, and then once it brings in the RNA polymerase, and the RNA polymerase is like all settled, ready to go, the sigma is gonna leave and then just kind of like hang around until it has somewhere else that it needs to do its job. But it doesn't like travel with the RNA polymerase, just to clarify. All right, so moving on specifically to gene regulation in prokaryotes, because again, we don't talk too much about eukaryotes. I think that was more like Bio285, I think, covered back then. But the main way that genes are regulated, or one of the main ways that genes are regulated in prokaryotes, is through operons. An operon is a set of genes transcribed under the control of a single promoter. So in this picture, you have a set of genes, and then you have one promoter. So one polymerase can basically transcribe a bunch of mRNA from just like one gene or set of genes. So having like this many different genes, it's called polycistronic mRNA, a single mRNA that codes for multiple proteins. Yeah, so essentially, if you want to think about it, you'll have like a promoter here. This is where the the RNA polymerase is going to bind, and then it's going to start transcribing. It's going to transcribe the entire set of genes, and then that's why it's like a single mRNA strand. But that single mRNA strand is going to code for 1, 2, 3 different proteins. So then you get transcription, you get this, like I said, one strand, 3 different proteins, and then the proteins happen through translation. Okay. Transcription of operons controlled by different transcription factors. So you can have activators or you can have repressors. The way that it sounds is the way that they work. If you're an activator, you increase transcription. If you're a repressor, they prevent transcription. And then activators or repressors can be turned on and off. Also, I forgot to say in the beginning because I always forget, but whether you're on Zoom or like in person, if there's ever anything confusing or you just have like a question that pops up, you don't have to like wait until the end or anything. You can just raise your hand or interrupt us. Okay. So for operons, to continue on that, each operon contains regulatory DNA sequences that act as binding sites for regulatory proteins. So every operon is going to have a promoter and a operator. Sometimes people get confused between an operon and an operator. An operon is like the umbrella term for the entire like system that's going on. An operator is part of an operon. The operator is where the repressors bind, whereas the promoter, like I said before, is where the RNA polymerase binds. So this little picture is here, the little purple, whatever shape that is on the promoter. That is the RNA polymerase. There's nothing on the operon. Sorry, there's nothing on the operator. See, I get confused too. Nothing on the operator, so there's nothing like blocking or repressing this operon from continuing. So your promoter can just go ahead and transcribe as needed. However, if you do have a repressor on the operator, then your polymerase cannot bind. And then it's, even if it could bind, it would be blocked by this huge thing that's blocking it. So that you would have no transcription. So those are the two like basic. Yeah.
Are there other operons or operators where activators can bind to it, or is it only repressors?
It can be activators or repressors. Okay. Either one, that's going to be like where the where the operator plays a part. Yeah. Kind of like down here, you can be inducible or repressible. So if you're repressible, repressors can bind onto it, but that's when your operon is always on. So if there's nothing on the operator, like it's just always going, and then something has to happen for it to turn off. If you're inducible, it's always off, and then something has to happen to turn it on. If you ever took 151 or you remember, that's like what the lac operon is. It's always off, but like if lactase is around, you want to try to like break it down so it turns on to break it down. The trip operon, which George is going to talk about in a second, the operon is always on because like the default is that you need tryptophan, but it can be turned off when tryptophan is around. So that's like an example of repression, but then this one would be like an example of activation. Does that answer your question a little bit, like the difference? Okay. Cool. So this is the generic trip operon, and then George is going to go into kind of like the specifics of like the different scenarios that can happen in the trip operon in a bit. Trip is like a short 3-letter word. It's an amino acid. It stands for tryptophan. When you have, there's like 2 different basic scenarios. If you have low tryptophan levels, that means that your bacteria or your prokaryote doesn't have access to tryptophan, but they want tryptophan, so they're going to try to make the proteins. And so you're going to want to turn the trip operon on. If you have high levels of tryptophan, you want to try to save energy, or like a cell always wants to save energy. That's kind of like the basic of life. And so you're not going to want to try to transcribe a bunch of things that you already have. And so if there's a lot of tryptophan around, the trip genes are going to be turned off. You can think about it as like, if you have a lot of like one item of clothing, you're going to want to save money, so you're not going to buy the same thing. If you have a shopping problem, that might not apply to you, but you can think of it that way. Okay, so a little question: is the trip repressor protein a positive or negative regulator of the operon? Yeah, go for it.
I want to say negative.
Do you want to explain why?
Because when there's a whole bunch, it's like punching you by not producing more. But when you have not enough, then it will be not enough.
Yeah, exactly. So my laptop is lagging, but it will say in a second that it is negative. I'm sorry.
Impressive.
I don't know why it's not. Yeah.
So I'm just curious. So is there other type of operants, or are you just using trip operant just like for this class?
For this class, we just because it'd be confusing to go over like a bunch of different operons. The reason I think Laura has talked about before that she uses trip operon is that it's a pretty like generic one, and it has like a lot of the aspects of like things that you could potentially see in various operons. So that's why she uses trip as an example, but there's like a bunch of different types. Yeah.
Okay, so we won't be able to know like other
No. You're gonna be responsible for a trip operon, and then that's about it. Sorry.
Also, I had a question.
Yeah.
About why it would be negative. Is it like similar to like negative feedback loop, where it's like if it's like doing the opposite thing, whereas like if it's low, it's on, whereas if it's high.
Kind of. So it'll make more sense probably the more we talk about the trip operon, but the reason why it's a negative regulator, hopefully to make it like as simple as possible. I'm sorry, what was your name?
Oh, right.
I'm sorry.
Right.
Bright. So what Bright was saying, when you have a lot of tryptophan, you don't want to be making tryptophan, right? So like that's the key right there. The repressor is usually not bound because like I said before, it's a repressible operon. So usually it's always on. You want to turn it off. So if you have a lot of tryptophan, then the tryptophan is going to bind to, actually I have a picture. I realize I have a picture. Okay. So if you have a lot of tryptophan around, so like say for example, you have like more than just one, but you have a lot of these around, you're going to be more likely to bind to the repressor. And if you bind to the repressor, that cues for it to go bind to the operator. And then when you bound to the operator, that makes the operon turn off. So that's why it's a negative regulator. Does that make sense? Like the flow of order? Okay. So yes, this is the overall trip operon diagram. If you ever want like a reference, I know Laura has different like diagrams, I think throughout her lecture slides and stuff. I like this because it's like a little bit more simplified. If you ever want to just see like what there is as a whole, it'll be on these slides and they'll be sent out too. So if you ever want like another reference or another point of view, you can go for it. Oh, sorry.
Sorry.
Yeah, go ahead.
For the research that a bunch of
do the activators also bind in there as well? Or do they bind in the part in front of the?
Oh, no, it would also be here. Yeah, it would, they will all go on the operator. Yeah. Just in this case, again, like for the class, it's a repressor. So yeah. Did you have another question?
Yeah, I was just wondering, I wanted to double check if the trip is always on.
Trip is always on, it gets turned off. Yeah,
Okay.
so it's repressible. Yeah. Any other questions? Is there anything else?
No.
Cool. All right, your turn.
We turn on the mic, right?
Oh yeah, I can turn mine off.
Yep. Okay. So in this scenario, there's a high level of tryptophan, in the cell. So in this case, what will happen to the operon, and will transcription occur? And if transcription occurs, then tryptophan will be made. So, yep. What do you guys think? Yeah, right.
Should be turned off.
Why do you say that?
Oh.
That's okay.
Well, since we already have a lot of tryptophan,
Mhm.
then they get more, so. The operator we turned off, transcription will not occur.
Mhm.
And
Okay, yeah, that's right. So what's going to happen is there's also tryptophan. It's going to bind to the repressor. So it binds to like random diffusion. If there's also tryptophan, it'll be a lot more likely that it'll bind to more repressors. So then the repressor will go to the operator, and they'll block the RNA polymerase from going down the strand. So then there'll be no initiation of transcription, and therefore there'll be less production of tryptophan. Yeah, good. Here's some review. So attenuation, this can be really confusing, but basically the way I like to think about it is like there's the four regions, there's 1, 2, 3, and 4. So if 2 and 3 occur, this means that transition can happen, but if 3 and 4 occur, this means that it'll be terminated. It'll be terminated, and this is like how it actually happens like, mechanically can be definitely really confusing. But so I think about it like on a timeline, there's like the polymerase is like going in and making the strand. At the same time, behind it, the ribosome is also going in and translating it as the ribosome, or as the RNA polymerase is going and transcribing it. So yeah, you see prokaryotes continue because they transcribe and translate simultaneously. So there's actually we'll talk about it later.
Yeah.
Yeah.
This is just kind of like what
Good review.
attenuation is. Like definition wise, attenuation means that it'll cut off early. Like suddenly it realizes that, oh, like we made a whoopsie. We don't actually want to follow through and like make all these proteins. So we have to find a way to like cut it off and like shut it down. So this is like the way that it works. Like definition wise, it's good to know like which ones are complementary. So 1 and 2 go together, 2 and 3 go together, and then 3 and 4 go together.
Yeah, and then the trip codons and stop codons are in, but yeah, I think we talk about that later.
Yes.
Later slide. Yeah, in this scenario, there's low levels of tryptophan. So in this case, what will happen to the operon? And it was just like last time, will transcription occur and will attenuation occur? What do you guys think? Sure. I'll go ahead.
I think transcription will occur because the opera will have to turn on.
Mhm.
In order to like get not repression, but the other thing.
Yeah, that's right. Yeah, just in general, even like without looking at this, if there's like a low level of tryptophan like around in the cell, then they're obviously gonna need it, so they're gonna make more of it. Or if it's the other way around, then they won't make it, but there's lots of tryptophan around. So it's just like the basic level. But yeah, so here there's no tryptophan, so the repressor won't bind. And yeah, the RNA polymerase can bind to promoter and start transcription. There's no repressor, and there also won't be any attenuation. Yeah, so in this case, the RNA polymerase, it goes to, let me think about how to explain this. So yeah, there's this region of the codon called the UGG. It's a trip region and trip codons in region 1. And so if it goes there and if there's lots of tryptophan around and tryptophan RNA, will come in and it'll keep on going. But in this case, since there isn't a lot of tryptophan, it'll stop there, and that will allow 2 and 3 to bind together because UGG is in region 1. And once 2 and 3 bind together, then because 2 and 3 leads to translation, that'll happen. While 3 can no longer bind to 4 because 3 is making a hairpin with 2. And that leads, so that's why transcription goes on. And then stop. Can be a little confusing. Do you guys have any questions on that?
There's gonna be like a summary slide at the
Yeah.
end too. I think that kind of gives you like basic information of like what you need to know, like when transcription can and can't happen. Yeah, so that'll help too at the end.
I was wondering like what the hairpin.
Hairpin? Oh, can you go back to like
Yeah. This one?
so a hairpin, this is you said it's color code, like this is the light blue, it's up there, that's a UGG, and that's a purple. And if they form a hairpin, they basically just form bonds with each other. Like forms
Yeah.
into a hairpin.
Like that structure is called a hairpin. I think it's also called like a T loop.
Oh yeah,
Like
T-loop,
it's like
yeah.
there's different names for it.
Yeah, you can just call it like a
hairpin.
loop, a hairpin. It doesn't matter on the exams or anything when you like write about it.
If you even just said like a loop forms 1 and 2 bind together, you don't even say loop. Like we get the gist of it. Like we know, like you
Mhm.
know that these go together, so it'll be okay.
Yeah, you don't need to remember the specific name or write it down in a cheat sheet or anything. That should be- Okay, were there any questions on this part specifically? Like why it keeps on going?
So basically because it's keeps going because 1 and 2, or which one's formed together in this scenario.
2 and 3 form together. So I like I said before, I think about it like on a timeline, the RNA polymerase and the ribosome are like both on the same strand at the same time. So this strand is being transcribed while it is being translated. So like when 2 and 3 form, the RNA polymerase is on section 4. It can keep on going. But if it was like let's say the other way around, if 3 and 4 form, then like if they form a hairpin, then the RNA polymerase can't stay on 4. It gets like yanked out physically, like it's pulled out of strand and it stops transcribing.
Oh yeah.
That makes
So
sense.
that would be like the first scenario in over.
That would be like if it didn't transcribe.
Yeah.
The attenuation, yeah,
Yeah.
that would be attenuation. Yeah.
Mhm.
Any questions on low levels and like the mechanism between why it happens? Just because I know like we say it basically and it's like, oh, if you have low levels, obviously you want to make more, so it's just gonna go. But I think sometimes for certain questions, she does specify like different regions pairing together. So you want to know that if 2 and 3 end up together, you're not gonna get attenuation because like George said, 4 is gonna be transcribed alone and then it's just gonna keep going.
Yeah.
So we want 1 and 2, 4 to transcribe and translate.
Yep.
And 2 and 3, it's gonna start to break apart. I'm correct or
What do you mean?
um. I'm just confused on the what 2 and 3 do and what 2 and 4 do. Because you said it's not transcribing at 2 and 3 or around 2 and 3.
So yeah, if 2 and 3 come together, that means that it keeps on transcribing. Well, if 3 and 4 come together, they stop.
So 1 and 2 keep them transcribed and translate, but 2 and 3 only transcribe.
Oh, 1 and 2 is just like when it pauses. It doesn't really like mean whether it stops or start or stops or keep going.
So it says here, like it lets the ribosome catch up. So if you think about it, like pretend the ribosome isn't there at first. You start with the RNA polymerase over here, right? And it starts transcribing, it's going, it's going. It makes the 1 region, it makes the 2 region. It keeps going. Once it has this leader region out, this is like the start codon. Once this is made and it comes like out of the RNA polymerase, that's when a ribosome can bind on, right? But you want to have a chance for like the ribosome to try to catch up to the RNA polymerase, and that's what like the 1 and 2 pause is about. Yeah. So then that's kind of the purpose of 1 and 2. And when 1 and 2 pause together, that allows the RNA polymerase to keep going and then transcribe 3. And then it's like at this point, the ribosome has caught up, sort of, but then it's going to get stuck on the 1 region because that's the trip codons. And it's going to be stuck because if there's no tryptophan around, it's not going to be able to get into the ribosome and like form the amino acid link. And so if it's stuck there, that means 2 and 3 are going to be free and able to bind together. And then that means that 4 is going to be able to be transcribed. And then it's just going to keep going. Yeah. Does that make sense a little bit more?
Anything.
Yeah.
Do you have a question?
So like we want the 1 and 2 pause, like the other ones are these you don't really want to try early termination to get stuff.
What depends on the circumstances of the cell.
Okay.
So you remember like before what Tina said, like if you have lots of tryptophan around, the cell won't want to waste energy making more tryptophan. Versus if there was no tryptophan around, the cell will like it will want to make tryptophan because there isn't a lot around to use. So.
You're always gonna have the 1-2 pause. Like that's always
Mhm.
just gonna be natural for the ribosome to catch up. Like that one's always gonna happen no matter what.
Yeah.
Unless you have like just straight up repression and like the operon isn't even on.
On the quiz this week and probably on the exams too, Laura's gonna ask you a lot of questions where like there's a mutation of like oh what if like the operator or like the repressor couldn't bind to the operator no matter what, and like how does it affect the cells like survivability when there's a low levels of tryptophan, high levels of tryptophan, and like that. So just like keep in mind like how it works, and yeah, that'll be good. Yeah, I think it should be fine. Yep. So in this case, it goes from low to high levels of tryptophan, and when this happens, what will happen to the operon? Will transcription occur and will attenuation occur? So if there are now high levels of tryptophan, what do you guys think?
Operon will get shut down.
Yeah, that's right.
It's like it made the mistake of making too much.
That's right. Nerve attenuation.
So transcription wouldn't occur.
It would start and then it would stop.
Stop.
Yeah.
Yeah, but it starts from low, so that means the repressor wouldn't bind yet. So then it'll start, but then later on, so we'll realize that oh, it actually is like a lot of tryptophan around. We'll need to keep on doing this, and it'll cut short. Yeah, attenuation will occur, and strength is cut short. That's good. Yeah. So at this time, since there is like tryptophan around, it goes past the trip codons here since there's tRNA. I mean the loop past it. But then on region 2, there's a UGA, it's another stop codon. This will if the ribosome gets to that point, it'll stop there like no matter what. It's just a stop codon. Doesn't matter whether there's tryptophan or not. And then so if the ribosome stops there, that stops 2 and 3 from forming a hairpin. So if 2 and 3 can't form a hairpin, that means that 3 is now or region 3 is now free to form the hairpin with region 4. And then, as you know, 3 and 4 from a hairpin, they'll yank the ribosome, or not the ribosome, the polymerase out of the transcript, and that will be attenuation. So they'll be yanked out before it can finish. So the whole thing won't be transcribed, and that means there will be less tryptophan. Yeah, what's up?
How does the ribosome get past the one?
What do you mean 1, 2?
Like over here.
Yeah, how does it get past that to get to the drop codon?
Oh, like the pause?
Yeah.
I think I'm remembering wrong, but I'm pretty sure it just goes through it and basically like flattens it out. That's how I remembered it.
Yeah, pretty much. So
Just
the way
runs through it.
I don't know if she still says this analogy, but she describes it as like a car crash essentially. You have the 1-2 together and they're complementary, so they're paired together at first, and then the ribosome is just there, like it's trying to catch up, like it's just going because there's nothing stopping it. So the ribosome starts at the AUG, it's just going and going. Once it reaches here, it literally just knocks into it, and then like they unbind. Yeah.
Yeah, because in this or in these scenarios, ribosomes only been stopped by like stop codons. It don't get stopped by a hairpin.
Yeah. So then that's why it'll stop at the stop codon.
Yeah, like even after yeah, that's right.
Yeah. After seeing this slide and like George explaining it, did like the last slide make sense a little bit more? Like the different scenarios between the two make sense between like just low and then low to high?
Yeah, it's okay if it doesn't make sense or if you want to ask more questions. This is a confusing part. No questions on Zoom. And that's the summary of what everything we went over. For situation where there are high levels of tryptophan, the repressor bound, so you don't even need to worry about attenuation. In the second level, when they're low, or second situation when there are low levels of tryptophan, the repressor doesn't bind, and 2 and 3 hairpin form, and then does full transcription and no attenuation. And situation 3, that we just went over, the ribosome stops at region 2 in the 3 and 4 form, which equals attenuation. Yeah. Are any questions about that? Or anything we went over?
Oh, okay. So that's technically the end of what we're covering for today. Like we said, like all the non-coding RNA stuff is going to be covered more on Thursday. Does anyone have questions in general about what we went over that they're like suddenly thought of, or problem set questions as well.
We'll probably start recording, right?
Yeah, I'm gonna start recording.
Any questions on